So welcome. Good afternoon or morning from where I'm at. This is part of the Early Researchers Seminar Series that's hosted by Cambridge Reproduction, SRI. We're very grateful to the volunteer coordinators for organizing this series and to all the speakers who have agreed to take part. And today we'll have two PhD students, including a visiting researcher, tell us about their work. The seminars will be held online on the third Thursday of each month at 1 p.m. Details of forthcoming seminars for next semester, as well as prior ones, can be found on the SRI website. Cambridge Reproduction is an interdisciplinary initiative that brings together researchers from across the university who have an interest in any aspect of reproduction, from science, technology, and medicine to arts and humanities. Membership is open to all staff and postgrad students, and gets you access to our events, funding schemes, and newsletters, which have highlights, events, and opportunities in reproduction research. You can download the membership form from our website and send it along to Christina Rosnek, who's been hosting us all term and is the Cambridge Reproduction Coordinator. So we're interested in receiving abstracts from students and postdocs for next term. Please let us know if you would be interested in participating. So our next presenter will be Antonia Hufnagel from the Institute of Metabolic Sciences, who's a PhD student within Cambridge.
Thanks everyone, and thank you very much for this nice organization of this nice seminar series. As Staci just said, I'm a PhD student in the lab from Professor Susan Ozanne in the Institute of Metabolic Science here in Cambridge. And in my lab, we really focus on the effect of obesity during pregnancy. And the reason for this is that in many populations, over half of the women are overweight or obese. And this means that more and more women also enter pregnancy obese. And we know from human and animal studies that maternal obesity does not only lead to complications during pregnancy, such as gestational diabetes, preeclampsia, preterm delivery, and even stillbirth, but it is also associated with increased risk for the offspring to develop cardiometabolic diseases later on in life, such as type 2 diabetes, hypertension, or obesity. And with the rise of women being pregnant with an obese or overweight weight, there's also an increased prevalence of GDM across the world. And nowadays, around 15% of pregnancies are complicated by GDM. So therefore, we need good treatment of GDM. So what I highlight here is just as an example, the UK NICE guidelines. And in the NICE guidelines, it's recommended that an oral GTT screening is performed at 24 to 28 weeks of gestation. And then if fasting or 2-hour glucose readings are above the threshold, then lifestyle and diet interventions are trialed. However, if these fail, then for a long time, insulin was used as the first-line treatment of GDM. But in recent years, metformin has been used more and more, and it's actually now also incorporated into the NICE guidelines. And the reason for this is that the advantages of metformin compared to insulin. As you probably know, insulin needs to be injected, but metformin can be taken as a tablet. Metformin does not need to be stored in the fridge, which is really good in developing countries. And it's associated with less hypoglycemic episodes, and metformin is in general cheaper compared to insulin. So therefore, it's not too surprising that more and more countries now use metformin in GDM treatment. This is not an exhaustive map I made here, but you can see with the orange countries that there are more and more countries across the world that are now using metformin in GDM treatment. Therefore, it is very important that we actually know and understand what metformin does during pregnancy. The initial studies of metformin during pregnancy come from women with polycystic ovary syndrome. In these women, metformin has been shown to improve fertility. Additionally, there are now studies that suggest that metformin can prevent preeclampsia, quite a detrimental pregnancy complication. And then there are studies in women with GDM where metformin improves glucose tolerance, and also studies in obese pregnancies without GDM where metformin can reduce weight gain. However, in recent years, there's been some studies on the long-term effects of metformin in pregnancy on the offspring. And these studies show that adiposity is increased in the offspring exposed to metformin during pregnancy. Where the data is very scarce when it comes to the effects on the fetus and on the placenta. And this is something that also a colleague from my lab has shown in a recent meta-analysis, because the only fetal kind of data she could find was birth weight reported in all these different studies. And what it was shown is that birth weight was reduced in those babies that were born to mothers with GDM who have been treated with metformin compared to those treated with insulin. However, when she then looked later in life, an infant weight at 1.5 to 2 years of age, weight was increased with metformin treatment compared to insulin treatment. And the same was true even later at 5 to 7 years of age when looking at the childhood BMI. And this is something that a former PhD student in my lab could also recapitulate in our mouse model. So what she showed was that 8-week offspring from mothers exposed to metformin during pregnancy had an increased fasted white adipose tissue mass. Interestingly, this was specifically seen in the male offspring. So all of this led to this study I will present to you today. So the aims of this study were to assess the impacts of metformin treatment in our mouse model in an obese glucose intolerant pregnancy on maternal physiology, but then specifically also given the scarcity of data on the placenta and the fetus. So in order to do this, we use our mouse model in which we feed our mice a chow or obesogenic diet from weaning onwards and then throughout the first pregnancy. And once the animals on the obesogenic diet have reached a body weight of over 35 grams, they are then mated for a second pregnancy. And this is the actual pregnancy that we're investigating. For the animals on the control diet, we just see that they stay below a body weight of 25 grams, and then they're also mated. As you can see, our high-fat diet is a high-fat diet plus condensed milk, which has a high sugar content, so that we can mimic a high-fat, high-sugar, so-called Western diet here. And then we have a third group, which is also fat and obesogenic diet. But once these animals have reached a body weight of over 35 grams, they're given metformin for 1 week prior to mating and then throughout pregnancy. And metformin is given in the condensed milk at 300 mg per kg per day. And this has been shown to be a clinically relevant dose as it equates to 1.7 gram of metformin in a 70 kilogram man. So this is clinically a clinical dose we would see in patients. When we look at the maternal phenotype, you can see, as you probably expect, that our animals on the obesogenic diet and those on the obesogenic diet treated with metformin both have an increased body weight throughout pregnancy when we compare them to the controls. We can also see on the right-hand side that the fat mass at the end of pregnancy at embryonic day 18.5 is increased in the obese dams. But interestingly, this can be reduced with metformin treatment. We additionally saw that the area under the curve in an intraperitoneal GTT, looking at glucose tolerance, was increased in the obese animals, along with fasting insulin levels. And both of these parameters, again, could be reduced and improved with metformin treatment. We also saw that our obese dams showed liver steatosis. And again, this was something that we could improve with metformin treatment as it reduced the percentage of fat in the liver. So all of this showed that metformin does improve maternal metabolic health overall. But as I said, we really wanted to focus a bit more on more pregnancy-related measures and on the fetus and the placenta. So for this, we take the same model I showed you in the beginning, and now we look at the end of pregnancy at embryonic day 18.5, which is just before the mouse would normally give birth. And at that time point, we perform ultrasound, we dissect the fetuses and the placentas to then weigh them and collect them for further analyses. This just gives you a little bit of an idea of how an ultrasound in the mouse looks like. So when you look at the arrow, you can see this is the blood rushing through the umbilical cord, which then attaches to the placenta, and you can see the body of the fetus and then likely a paw from the fetus looking out. But what we used our ultrasound mainly for was to locate the uterine artery. So we could locate the uterine artery branching off the iliac artery at the height of the bladder of the mouse. And then we see this very typical waveform of the uterine artery. And this allows us to measure the systolic peak, the diastolic velocity, minimum diastolic velocity, and the mean velocity in the vessel. And all these measures then allow us to calculate the pulsatility and the resistance index. And both these indices are really measures for the resistance in the vessel. So we know from human pregnancy that with advancing gestation, these indices are decreasing, meaning that also the resistance in the vessel is decreasing, meaning that there's more insufficient blood supply to the placenta and the fetus. But when we looked at these indices in our obese dams, we could see that they were increased compared to the control animals, highlighting impaired uterine artery compliance in these animals. When we then looked at metformin treatment, we could see that we could rescue this phenotype, and these indices were reduced back to control levels with metformin treatment. Interestingly, we could see that fasting insulin levels in the mothers predicted uterine artery pulsatility index. So this impaired uterine artery function really made us think of a preeclampsia-like phenotype. As I said in the beginning, preeclampsia is a very detrimental pregnancy complication, and one hallmark of it is impaired uterine artery function. Other symptoms in the clinic are, for example, proteinuria or hypertension. In the clinic, soluble FLT, S-FLT, is used as a typical biomarker. S-FLT is thought to also lead to the angiogenic dysbalance we see in preeclampsia, as it binds vascular endothelial growth factor. So what we did was to measure S-FLT in the serum of our dams, and we could see that was increased in the obese animals and reduced again with metformin treatment. But the big question now is really, what does all of this now mean for the fetus? When we look at the fetuses, we see that in line with the reduced and impaired uterine artery function, fetal body weight is reduced in the obese group. However, despite all the improvements we can see with metformin in regards to maternal metabolic health and the improved uterine artery function, metformin does not rescue fetal body weight here. When we look at placenta weight, we can see that placenta weight is increased in the obese placentas, and there's no difference when we look at metformin placentas compared to controls. We can now take the fetal body weight and the placenta weight to calculate the body weight to placental ratio. And this is a measure for placental efficiency in mouse models. And we can see that placental efficiency is reduced in the obese group. And again, metformin does not rescue this. You can see on this slide and also on the previous slides that we always look at male and female fetuses and placentas separately, because I now feel there's now growing interest into sex differences. So they've been shown to be quite important. And you can see here that it seems like the female placentas are slightly more efficient throughout all the groups. But this finding really led us to think more about the placenta and ask us what is happening with these placentas, why are they less efficient? In order to look at this, I performed histological analyses of the placentas. So I stained the placentas for the outer trophoblast layer of the placenta, and then I use a marker for endothelial cells, as you can see on the bottom, which marks the labyrinth layer of it. So I can nicely delineate the labyrinthine zone of the mouse placenta. And this is the zone where the key exchange zone for the exchange between maternal and fetal blood. And I can see that this labyrinthine zone is reduced in the obese animals. And again, this is a phenotype not rescued with metformin treatment. When I looked at the sections for this of my placentas, I could see weird black dots all around in those obese and metformin-treated placentas. When I then stained them for H&E, I could see that weird knots appeared in these areas. And with the help of a histopathologist, we were able to show that these dots were calcium. So you can see here that there is increased placental calcification in the obese placenta, which you can't see in the control placenta. And again, this is something that was not rescued with metformin treatment. We were quite interested in this placental calcification phenotype. So for that reason, I took obese placentas at different gestational ages. And as you can see here, we could show that there's no placental calcification earlier in pregnancy at E13.5, but it starts to emerge at E15.5 and is then present at E18.5. And what we additionally did is to locate what is happening in those areas where we see calcium. So these are serial sections. You can see where we have calcium. We saw some overlap with apoptosis staining. We also saw quite a striking overlap actually with areas of macrophage staining. And when we then look at the endothelial cell staining again, you can see that in those areas where we see calcium, this typical labyrinthine pattern seems to be disrupted. This placental calcification is also quite interesting in regards to human pregnancies, because calcification is a normal aging sign of the human placenta. So it's normal that it happens towards term. However, if it happens preterm, so before week 36, it is critical, and it's a sign of fetal growth restriction and stillbirth. And interesting for this study, it is associated to obesity, GDM, and preeclampsia. So therefore, this really shows that our mouse model resembles phenotypes we see in human obese pregnancies. If I now put everything together I've told you so far, we can say that our study definitely adds evidence that metformin might indeed be able to prevent or treat a preeclampsia-like phenotypes. We also show that indeed metformin does improve maternal metabolic health. However, despite all these improvements, we see that metformin does not rescue the reduced fetal growth, and it does not rescue any of the placental pathologies we see in the obese group. So therefore, we hypothesized whether there might be direct effects of metformin on the fetus and the placenta. In order to look at this further, we measured metformin concentrations, and we could see that metformin was present in the placenta, in the fetal kidney, in the fetal liver, and amniotic fluid. Amniotic fluid is interesting as the fetus repeatedly swallows the amniotic fluid, so might be exposed to metformin repeatedly. When you then look at the correlation between maternal and fetal serum, you can see that there's a strong correlation. And when you look at the values for fetal serum and maternal serum concentrations, we can see that metformin in the fetal serum is present in similar concentrations compared to the mother, even a bit higher. So therefore, we can definitely say that metformin does enter the fetal circulation. We can also confirm this when we looked at metformin transporter expression. So these are just raw CT values. So a low CT value marked in red shows that we have expression there. And we can see that there are metformin transporters in the placenta and in the fetal kidney, and also to some extent in the fetal liver. So therefore, the ongoing work now really needs to dissect out whether there are direct effects of metformin on the fetus and or the placenta. So metformin is thought to act via many different pathways. So when you look in the literature, there are many proposed pathways via which metformin can act. However, the key pathway that's always mentioned is that metformin can block complex 1 activity in the mitochondria and thereby activate AMPK. And AMPK obviously has loads of downstream effects. So it's known to inhibit mTOR, an important growth signaling protein. It's known to inhibit gluconeogenesis and lipid synthesis. So all of this could really alter placental and fetal metabolism and thereby impair or at least affect fetal development and growth. So in order to look into this further, we want to look at known targets of metformin. So for example, by assessing mitochondrial function in placenta and fetal tissue. We then also started looking at AMPK and mTOR signaling in the placenta and the fetus. We looked at GDF15 in the placenta because GDF15 has been recently shown to be a key mediator of metformin action. We could see that GDF15 was increased in the obese placentas, but not further increased with metformin. So there doesn't seem to be a direct effect of metformin here. And then we decided, given the scarcity of data around metformin effects on the fetus and the placenta, and given all these different pathways for metformin that are possible, we decided to start a very explorative approach and perform lipidomic and RNA-seq analysis of the placentas. So we now put everything together I've shown you in this mouse study, but also based on all the human observations we know, there are definitely benefits for the mother. So metformin has immediate benefits for the mother as it improves glucose tolerance and reduces preeclampsia risk. It then has practical benefits as it can be taken as a tablet and can be stored easily. And there are long-term benefits of metformin as well. So there are a few animal studies that show that there might be improved postpartum metabolic health in the mums. And there are also the suggestion that there's reduced interpregnancy weight gain, which is then obviously beneficial for subsequent pregnancies of this mother. However, on the other hand, we know from this study now, but also from some human observations, that high metformin levels enter the placenta and the fetus. So we can't exclude that there are direct effects on the fetus. And some of these direct effects could then mediate the long-term effects I've told you about in the beginning, such as that we see increased adiposity later in life in the offspring. So this is why metformin in pregnancy is a complex balance. And the big overarching question in the field is probably whether we can maintain all these very beneficial effects for the mum, but minimize fetal exposure. And this is something my lab will be looking into in the future by looking at different metformin treatment regimes and metformin formulations. And with this, I would like to thank my whole lab for creating such a lovely lab atmosphere all the time and for all their help with this study. And then all the collaborators and the people in course in the Institute that helped along the way. And I'm also very happy to take any questions.